Thomas Hunt Morgan was an American scientist who made important discoveries about how traits are passed down from one generation to another. His work in the field of genetics changed the way scientists understood heredity, and his discoveries continue to be important in modern science today. Morgan's studies on genetics, particularly his work with fruit flies, helped to uncover many secrets about how traits are inherited, and these findings laid the foundation for the field of genetics. Morgan's groundbreaking research was recognized worldwide, earning him the Nobel Prize in Physiology or Medicine in 1933 for his discoveries on the role of chromosomes in heredity. Morgan was born on September 25, 1866, in Lexington, Kentucky, in the United States. He grew up with an early interest in nature and science, a curiosity that would shape his entire career. From a young age, Morgan loved learning about how living things grow and change, and he was particularly fascinated by the questions surrounding heredity and how offspring resemble their parents. He later attended the University of Kentucky and earned a degree in biology, before continuing his studies at Johns Hopkins University, where he would eventually carry out much of his future research. In the late 1800s, scientists were still trying to understand how traits, like eye color or hair color, were passed down through generations. At the time, no one fully understood the process of inheritance, and various theories had been proposed to explain how children inherited traits from their parents. Some scientists believed in the theory of blending inheritance, which suggested that the traits of two parents would mix together to form the traits of their children. Others believed in a more complex idea that traits were controlled by gemmules, tiny particles inside the body that carried information from one generation to the next. However, none of these ideas could explain the actual mechanisms behind heredity, and scientists were still searching for a more accurate theory. Thomas Hunt Morgan, as a young scientist, became fascinated by the work of Gregor Mendel, a scientist from the 19th century, who had studied how traits were passed down in pea plants. Mendel's experiments showed that traits were inherited according to specific patterns, but his work was not widely recognized until after his death. Morgan, however, recognized the importance of Mendel's work and decided to apply the same principles to animals. He believed that by studying how traits were inherited in different organisms, he could uncover a more complete understanding of genetics. Morgan chose to study fruit flies, which were ideal for experiments due to their short life cycles, simple genetics, and small size. Fruit flies, scientifically known as Drosophila melanogaster, were easy to breed and observe. They reproduce quickly, with each generation taking only about 2 weeks to develop from egg to adult. Fruit flies also have only 4 pairs of chromosomes, which made them an ideal subject for genetic research. Morgan's first experiments with fruit flies focused on studying how various traits, such as eye color and wing shape, were passed from one generation to the next. His work would lead to important discoveries about the structure of chromosomes and the way traits are inherited. In 1910, Morgan made a key discovery that would forever change the field of genetics. He found that the gene responsible for eye color in fruit flies was located on the X chromosome, one of the 2 sex chromosomes. This was a crucial discovery because it provided evidence that chromosomes carried the genetic material responsible for inheritance. At the time, the idea that chromosomes carried genetic information was not widely accepted, and Morgan's findings provided strong evidence for the chromosome theory of inheritance. Morgan's research helped to confirm that genes are located on chromosomes, and that these genes control how traits are passed down through generations. Morgan's research with fruit flies also led to the discovery of genetic recombination, a process in which genes on the same chromosome can be shuffled during reproduction. He observed that genes located close to each other on a chromosome tend to be inherited together, while genes that are farther apart have a greater chance of being separated during reproduction. This idea, known as genetic linkage, helps scientists understand how traits can be inherited in patterns that are not always straightforward. Morgan's discovery of genetic recombination was one of the key pieces of evidence that helped to explain how genetic variation occurs. As Morgan's research gained more attention, other scientists followed his lead and began studying genetics using fruit flies. Morgan's work helped establish the field of modern genetics, and his experiments with Drosophila became a standard for genetic research in the future. His discoveries provided a foundation for scientists to study the inheritance of more complex traits, and they contributed to the understanding of genetic diseases. By studying fruit flies, scientists could now better understand how different genes interact with each other, and how they influence traits in living organisms. Morgan's work also had an impact on the study of evolution. He showed that changes in genes could lead to new traits, and these changes could be passed down to future generations. Morgan's research helped scientists understand how genetic variation leads to the diversity of life on Earth, and how species evolve over time. His discoveries contributed to the broader field of evolutionary biology, and helped to explain how new species might arise through natural selection. In 1933, Morgan was awarded the Nobel Prize in Physiology or Medicine for his discoveries on the role of chromosomes in heredity. This recognition was a testament to the importance of his work in shaping the field of genetics. By the time he received the Nobel Prize, Morgan had already become a leading figure in the scientific community, and his research had opened up new possibilities for the study of genetics, evolution, and medicine. Morgan's discoveries not only revolutionized the understanding of heredity, but also paved the way for future research in genetics and molecular biology. Morgan continued his research and teaching throughout his life. He became a professor at Columbia University, where he mentored many other scientists who would go on to make their own contributions to the field of genetics. Many of his students, including scientists such as Alfred Sturtevant and Hermann Muller, went on to make important discoveries in genetics, building on the foundation that Morgan had established. Morgan's influence extended beyond his own work, as he helped to shape the future direction of genetic research. In addition to his research on genetics, Morgan also made contributions to the study of the behavior of chromosomes during cell division. He discovered how chromosomes move during the process of meiosis, the type of cell division that produces reproductive cells or gametes. His work on meiosis provided important insights into how the number of chromosomes is reduced in gametes, and how the chromosomes from each parent combine during fertilization. This understanding of cell division helped explain how traits are passed down in a more detailed and accurate way. Morgan's work also had important implications for the study of human genetics. His research on the inheritance of sex-linked traits, such as color blindness, helped scientists understand how certain genetic disorders are inherited in humans. Morgan's discoveries made it clear that many genetic traits are linked to the sex chromosomes, which was a breakthrough in the study of human diseases. His work provided the foundation for the study of genetic diseases, and helped lead to advances in medical genetics, including the development of genetic counseling and testing. Thomas Hunt Morgan died on December 4, 1945, but his legacy continues to shape the field of genetics today. His pioneering work with fruit flies opened the door to the study of genetics and molecular biology, and his discoveries remain central to our understanding of heredity and evolution. Morgan's contributions to science have had a lasting impact on biology, medicine, and many other fields, and his discoveries continue to inspire future generations of scientists. Through his work, Morgan helped to uncover the secrets of inheritance and genetics, providing answers to some of the most fundamental questions in biology. Without Morgan's groundbreaking discoveries, the field of genetics would not be what it is today. His research paved the way for new medical treatments and helped scientists develop a deeper understanding of how living organisms inherit traits and evolve over time. Morgan's work also opened the door for future research into the genetic basis of diseases and the potential for using genetic information in medicine. As a result of his work, the study of genetics has continued to grow and expand, and Morgan remains one of the most important figures in the history of science.